speaker for this afternoon session. I would like to again thank Dr. Purabi for a very nice presentation and for answering the questions from our participant. So this last presentation from Ms. Nur Hikmah, her title is Transient Expression of Meloidogyne incognitas, 16D10 gene in planta for banana, 16D10 protein-protein interaction via affinity tagging purification-mass spectrometry, APMS approach. Okay, very tongue twister title. So I would like to invite Ms. Nur Hikmah to share your slides and begin your presentation.
All right, thank you so much. I will share my screen now. Can you guys see the screen?
Yeah.
All right. Okay. Assalamualaikum and good afternoon, everyone. I'm glad to have this opportunity to present about my research, which is about banana nematode interaction, transient expression of Meloidogyne incognita, 16D10 gene in planta. Right. So my presentation would first about introduction, and then the objectives, and also I will explain the workflow of this research. And based on this workflow, I will explain the materials and methods, results in discussion, and also the conclusions. So introduction about bananas. I don't think I have to emphasize more about how important banana is, right? But for my presentation, for my research, we are focusing more on the plant parasitic nematodes that cause 30% of banana yield loss. So what are plant parasitic nematodes? They are small worms and they infect several plants and crops. Then they could cause a damage of up to 100 billion USD per year. And root knot nematode is one of the plant parasitic nematodes. And in this research, we focuses on Meloidogyne incognita, which is the most dominant species that infect banana plants in Peninsula Malaysia. And typical symptoms of plants infected by this nematode are root galls, wilting, and stunted growth. And all of this could weaken the root system, and the plant could topple over before it can be harvested. And as you can see in the picture, the root galls. So more on Meloidogyne incognita, they are obligate sedentary endoparasites. And during their juvenile stage 2, they would penetrate the root tissues and establish their feeding site. So what's important characteristic of this nematode is that they have dagger-like stylet, and this could penetrate the plant cells physically, and this opens up opportunity for secondary pathogen infections such as fungi and viruses. And they also have subventral gland that produce the effector molecules, and these molecules are the ones that could manipulate plant system to induce the giant cells and serve as the nutrient sink, as you can see in the root galls in the roots. And the nematodes are pathogens that secrete effector molecules that can target multiple layers of the plant immune system, PTI and ETI. And incompatible interaction, effector proteins or PMP would be recognized by the plants, and this would lead to their defense mechanism, and they could successfully evade the pathogen infection. But in a compatible interaction, the pathogen would successfully manipulate the plant's immune system, and this could cause diseases and parasitism in the plants. Right. So about 16D10, in 2003, one group have discovered 37 cDNA clones that are secreted by this nematode during their parasitic life cycle. And one of them is the 16D10 parasitism protein. So this parasitism protein was shown to interact with the host regulatory proteins and induce the enlarged multinucleated cells. And 16D10 is actually a small peptide with 30 amino acids of signaling peptide and 13 amino acids of mature peptide. Right. So it is shown that when they overexpress this 16D10 gene in Arabidopsis, you can see that this protein interact directly with the host regulatory protein and induce root proliferation. So we are trying to capture the 16D10 and plant protein interaction. So we come up with the APMS approach. So APMS approach had been used to study protein-protein interaction between human and HIV, and also protein-protein interaction in Arabidopsis and other plants. But this is actually the first time that APMS approach would be used to study plant and pathogen interaction. So explaining more about this approach is that first, we need to express the bait protein in plant tissues. For example, here we use 16D10, and we hypothesize that this bait protein would then interact with the plant proteins, and this would form a protein complex. Thus, this protein complex could then be purified in the affinity column, and then we can identify this protein complex using mass spectrometry. And lastly, we want a result that could identify the protein-protein interaction network of this 16D10 with plant proteins. So the problem statement here is that there's no banana varieties that are resistant against the Meloidogyne incognita, and the management like crop rotation and nematicides, they are not sustainable and harmful, and therefore we would like to understand the molecular mechanism of the interaction between the pathogen and banana, so we can come up with a better solution or a better banana in the future. So the objective is to reveal the banana proteins that are interacting with this 16D10 gene using APMS approach. So the hypothesis is that the transient expression of this 16D10 in the plant tissues could allow the plant proteins to form a complex, and this complex could then be pulled out and characterized using APMS. So my research have 4 workflows. I will explain them stage by stage. So the first workflow of my research is to construct the expression cassettes. So we designed and constructed 4 expression cassettes, and we cloned them, and we also sequenced them to confirm the orientation of the cassettes. So we successfully clone and put the expression cassettes into Picambia 1304, and these cassettes would then subject to transformation into Agrobacterium. So the next part is transforming of these expression cassettes into Agrobacterium and see their expression. So this includes protein extraction and SES page. And from the 4 constructs, there's only 2 of them that we can detect using Western blot. And both of them are the matured 16-10 peptide. And this is similar to what Huang had discovered too. And this could be because of the signaling peptide that affect stability, folding synthesis, and protein biosynthesis of the protein. And we can see that this construct showed more efficient detection in Western blot. So we choose this construct for further investigations. We also did a spiking experiment, which is that we mix Agrobacterium protein and the banana roots protein, and then we mix them in a ratio. This is to detect how much of this recombinant protein that we need for detection in Western blot, because this is a heterologous transient expression. And based on our result, we need 9 milligram per mil of concentration to be detected in the Western blot. So this is actually a challenge for us to optimize the transformation method in plants, right? So the next part of this study is to express construct in the banana root tissues using Agrobacterium, right? So actually, there's no optimized protocol available yet for transient expression in banana root tissues. Therefore, we have to design our own Agrobacterium-mediated transformation in the roots to mimic the 16E10 and plant protein interactions. And we designed 2 different methods. The first method, we grow the banana in soil and transfer the agrobag using scalpel. The second method, we grow the bananas in hydroponic and then inject the agrobacterium with syringe. So the first method is adapted from the single inoculation method. And what we did is we just scar the roots and then we treat them with agrobag containing the expression vectors. And the transformation method 2, we design our own hydroponic system. And then we also use the syringe to transform the agrobacterium into the banana roots. Right, so these are the results for the method 1. So as you can see in method 1, we did get some transformed cells, but this is not enough for detection in Western blot. As you can see in our previous spiking experiment, we need more amount of recombinant protein. But in the method 2, with the hydroponic grown banana roots, you can see that we have a high amount of gas stain or transformed roots of the bananas with different time points. So in summary, we have a good gas staining visibility up to the 5th day, but the quality of the harvested root tissues on the 5th day, they started to wilt. So based on the gas assay results, we plot the graph and we see that yes, the hydroponic grown methods are more efficient than the soil grown. And from the hydroponic grown methods, there's the time points. We can see that although 5 days have the highest transform roots, but we actually recommend to harvest them at 3 dpi with 81% of transformation capacity. This is to obtain better protein yield while maintaining the high amount of gene expression. So yeah, I guess we can say that we successfully optimized a transient expression method for heterologous gene expression in banana roots. And I hope that this method could also be adopted for other non-model plants. Right. So the last part of this study is the APMS. And so the first part is of affinity purification, where we extract the plant protein transform and purify them in a column, and then use SDS page and Western blot. So based on the SDS and Western blot results, we can see that we detected the 98 kilodalton protein band of the recombinant protein. And also in the purified protein length of the SDS page, we can see that there's several bands of 16 to 175 kilodalton other bands. And this could indicate that the presence of the putative banana root proteins that are interacting with the 16D10 and that are successfully pulled out during the affinity column. So what we did is that we cut the gel plots of the SDS page and we prepare them like trypsin digestion and desalting and send them for LCMS to identify the peptides. Right. So now we have the peptide identification based on the LCMS data with their own function and annexation number. And here we have 23 plant proteins that are the possible interacting with the M1610. So yeah, in 2017, Al-Idris have also detected plant proteins that are involved in this nematode and banana interaction. And some of the proteins include the calcium binding protein, PR proteins, and LOX protein. So based on literatures and our LCMS data, there are possible protein-protein interactions that are of interest, that which are binding proteins. And because this parasitism gene would target the plant immunity, such as ROS that induce the PTI. And they could also target the plant cell programming by modulating the gene expression. For example, the GLAN4 effector protein from cis nematode, they had been shown to have a DNA binding ability, and they can downregulate a defense gene. Another possible protein-protein interactions are with the kinase activity protein, as Castaneda had showed that upregulation of the LR protein kinase family are detected in the transcript of banana inoculated with M. incognita, and also hypothesized in Al-Idris. And another possible protein-protein interactions are with chloroplast complex, because the modifications of chloroplast could be related to plant hormones that are involved in ROS production. And lastly, the cell cycle regulation proteins, because nematode effector would target the cell cycle and cytoskeletal components that could lead to the formation of the giant cells. And these proteins involved are also detected in Al-Idris paper. So in conclusion, expression of the TEC16D10 protein in plant tissues allow the interacting complex to bind, and this complex could be pulled out in the purification step and then identified in mass spectrometry. So yes, we successfully showed that APMS approach could be applied to study protein-protein interaction between banana and 16D10 of M. incognita. Therefore, our future work, we need to validate these interactions with yeast to hybrid or biopsy, and to also confirm the interactions and build a protein-protein interaction network. So these are my references and acknowledgements and fundings of the research. Thank you.
Thank you so much, Ms. Nur Hikmah Mostaffa, for very detailed, informative presentation. We are really running out of time, but I think I can read only one question posted in a chat box. So by Dr. Paul Dennis, very interesting presentation. So could you use APMS to identify proteins association with plant response to FOC? Like instead of
Yes.
your Meloidogyne?
Yes. Actually, we can use this approach to study the FOC genes too. Yeah. But since this is like the first time this approach is used in bananas and in plant pathogen, so yeah, we tried it with 16-10 first. But in the future, yeah, maybe.
Yeah, we can try to use that and do a study on that, right?
Yeah.
Okay. Yeah. Is there any question from participants? Direct question. If I can take one, maybe. I think the plenary session in our main hall started, I guess. So with that final presentation by Ms. Nur Hikmah, I think we can end this wonderful afternoon session